Welcome to the ASN Kidney Week podcast, exploring the latest advancements, research, and trends shaping the future of kidney care. Tune in for expert conversations, clinical insights, and highlights from ASN Kidney Week, a go-to source for staying informed and inspired.
Hello, everybody. I'm Matthias Kretzler from the University of Michigan. I'm professor of internal medicine and computational medicine and bioinformatics, and I am the program committee co-chair with Kathy Tuttle.
Hello, everybody. My name is Katalin Susztak. I am a physician, scientist, nephrologist at the University of Pennsylvania, and I am a professor of medicine and genetics at the same institution.
Hello, my name is Julio Saez-Rodriguez. I'm the head of research at the European Bioinformatics Institute in Cambridge, UK. I'm Professor Emeritus of Medicine in Heidelberg University, and at the same time as an IgA nephropathy patient.
Welcome, everybody, to our podcast. It is such an exciting time in nephrology, and we were all thrilled to hear from our president yesterday in the opening plenary to hear where nephrology has progressed. After decades of stagnation, we finally have what Prabir referred to as a substrate established by a lot of basic science for many, many years from many of us around this table here, that substrate finally is leading to growth of impactful clinical trials anchored in a deep understanding of kidney disease. And hopefully you are seeing throughout Kidney Week in all the different sessions that common thread, how we learn how kidneys work and fail, and how we take that knowledge now excitingly forward to identify novel ways to interfere with kidney failure, keep kidneys and people alive. And excitingly, we are learning now from these studies how and where we can do even better. And it's really exciting to have two leaders in the field around their table who will give us their perspective where and how the progress has developed. You might have listened or seen the session this morning where we were very excited to honor Dr. Katalin Susztak with the Homer Smith Award, the preeminent award the American Society of Nephrology gives out. You have seen her groundbreaking work where she established that substrate where so many other studies are coming from. And I was personally moved when Dr. Saez-Rodriguez, the director of the European Bioinformatics Institute, the largest bioinformatic research unit in Europe, and truly at the cutting edge of how we learn with an informatic perspective how cells work and talk to each other. When Julio said to us that he is not only on the forefront of research as a scientist, but also on the forefront of research as the patients in the trial, where the day before we heard the outcome of the trial, and that the trial has cut down the disease burden by more than 2/3 and has a true opportunity for the very first time to stop a disease process entirely. So we would love to hear flavors from each one of you, what you find exciting about Kidney Week, where you think science is going, and where our listeners maybe want to engage in this endeavor going forward. Katalin?
This has been an amazing meeting, starting with the presidential address, which was truly uplifting this year, starting from the 1990s when we had ACE inhibitors to 2025 when we have more patients and more trials and more patients in trials than excuses that we had previously. My interest is common kidney disease development, such as looking at diabetes and hypertension attributed kidney disease, which represents roughly about 75% of all patients with kidney disease worldwide, and that could be maybe 600 million people. The approach that I feel has been really powerful in our field is mapping the disease, starting from genetic mapping, working through mechanism, and those approaches led to multiple new medication development. The session yesterday by Vlado Perkovic was really uplifting. I was there in 2019 when Vlado presented the results of Credence in Melbourne at this particular nephrology venue, which was the first truly positive clinical trial showing that SGLT2 inhibitors has a remarkable effect of protecting from GFR decline. That was followed by other drugs such as the GLPs, the MRAs. Now we have almost an entire alphabet soup for our patients. And also, these medications actually are in the television as advertisements. We are actually directly advertising for patients with kidney disease. This is a truly remarkable milestone, as Vlado actually highlighted. Based on that, this meeting was really showing the power of basic discovery and basic research. How we are essentially discovering more genetic variations that explain kidney function heritability, which is actually quite strong. How we could use variety of omics technologies, and I think you have heard so much single cell, so much spatial omics that your head may be spinning by this time, but I think those approaches with millions of cells are helping us to really define how each cell is behaving in the context of diabetes and common CKD. Lots of new pathways, lots of potential targets have been uncovered by large consortia such as KPMP and many other individual labs, the European consortia and other labs. Those will be the substrate for future therapies development. This is a truly exciting time, and I think our next challenge is going to be to put all this together using modern bioinformatics and AI approaches, such as by Julio, probably going to tell us how we're going to find the true target cell type and the true genes for all these diseases.
Yeah, thank you, Katalin. And indeed, coming to this meeting is an extremely energizing possibility. I'm a relative newcomer to the field. So when I come here, I really feel I'm overwhelmed by the advances, by the enthusiasm of the community. I'm coming here with two hats, as I mentioned before. One is as a bioinformatician, and the other one is as a patient. And in the first dimension, following what Katalin said, it is indeed really exciting to see all the progress, all the data, and all the expertise. And in the plenary we had this morning, where we awarded to Katalin and also to Dr. Rafael Kraman for their progress in the field. It was really great to see all the things that are happening, but the great opportunities ahead. And this is for people in my community, I think, a great motivation and a great opportunity to contribute. The kidney is a fantastic organ. It's a very complex system. As Matthias Kretzler likes to say, the most complex organ, perhaps only matched by the brain. And I think there is a lot of exciting challenges where the computational community can contribute, and together with the people that are leading the field, like Matthias, Katalin, and many others. At the same time now, putting my second hat as a patient, it's extremely unique opportunity to be here and see the advancements. As Matthias said before, we heard about all the new trials, and yesterday when Dr. Richard Lafayette shared the outcome of the Origin 3 is really unique to think how my life may have changed now for the better in a way that I could not have expected. So how powerful is science, and how powerful is the community in bringing forward. And then walking even across the posters to see all these new trials, all the opportunities, all the studies that make us have a big hope for me, but also for all the other patients that have IgA nephropathy, but also that they have other kidney conditions, that they can really finally look at the future with a lot of hope and really the opportunity to improve our cases.
And we really want to send that message loud and clear, particularly to the trainees, the junior clinician, junior scientists. There never has been a better time to be in nephrology. The opportunities you have in front of you are unprecedented. We around this table, we have committed a lifetime of effort to get us into a space where now you can start connect dots we even didn't imagine were present when we started our careers, with an approach that your impact will change the lives of people today or tomorrow. And wherever you are and wherever your research interest is, there is a rich framework of knowledge, of colleagues, actually of friends available to connect and to change our way we treat people with kidney disease. So despite the fact that, yes, we have a lot of background noise happening right now, Never has been a better time to be in kidney, and we encourage you connect with each and every one of us you see in front of the posters at meetings, at symposia. If you have an idea, if you want to engage, there are many different ways. You saw that the ASN has built up tremendous resources to support scientists in all their different aspects of their career to move forward with their research programs. So please feel free to reach out in ways we can help you to engage, and we can bring our insights closer to patients.
Absolutely. I think this is the goal, as Matthias says, but I think also we want to highlight that Matthias spent his entire career making all this data publicly available. I think we have also a website that we host. There are several others by HubMap, by the Humphreys Lab, and many others, where you can look at the data, you can look at your favorite cell type, you can look at your favorite gene, and just see whether the association is present or expressed in a specific circumstances. So even if you're not interested or unable to do high-level computational analysis, these data sets are out there and easily available and downloadable. These are why these resources have been created. But I think I want to pose different questions to this group. How do we solve the major bioinformatics challenge? I think if you're a clinician, your head might have been spinning with listening to all these talks about all the millions of cell types that we individually profiled, and we cannot get to know all the million cell types personally. So how are we gonna make sense about this newfound enormous information set?
I think this is indeed an important challenge, but one we should be working on, and we should try to leverage as much as we can. On one hand, of course, of the expertise of all the community, and on the other hand, these new upcoming AI tools, like you have done pioneer work with this foundation model of the kidney. There are all these now agents that can allow to give us easier interface to the knowledge, and these agentic frameworks allow us to tune the level of complexity to different users. There is still a lot of work to be done. We need to do it in a correct way, in a responsible way, that they give information that is reliable and trustable. But I think combining the experts, so all of you in this community, together with these new tools, I think we can then, and we agree, this is something we need to address, how to make all this knowledge accessible for all of us.
And really, let's keep that knowledge cycle going. So we started out in observing how kidneys work and fail, but now with the clinical trials, among the entire aspect of these different facets of kidney disease, we can capture how trial intervention help some people and not others. And that gives us an opportunity then to bring that insight back and use the knowledge environments available to identify innovative strategies, how to close the treatment gaps who remain with our patients. And again, Vlado's presentation yesterday morning showed it beautifully, how step after step we are bending the curve of GFR loss upwards so that we have an opportunity to approach on a population level an area where we are close to preventing kidney failure loss in the patients. But don't be fooled, there will be many patients who still will lose kidney function, and where we will have to use the tools to identify distinct mechanisms of actions who drive diseases forward and lead to kidney function. For example, Katalin, with your genetic catalog, I think that is a fantastic way to define where an individual starts her or his life and how gene-environment interactions intersect. Do you want to give us an introduction how this tool has changed the way we do drug discovery today?
Oh, well, yes. Thank you so much for highlighting this one. It's been a really pleasure to try to work on these tools that essentially translate these basic discoveries to try to make it clinically usable for the clinicians. And if anybody has suggestions, we would be very happy to see. What we did in our recent studies, we mapped the genetic architecture in 2.5 million people, And that identified 1,000 loci. And from that 1,000 loci, we used 32 of these different single cell and omics tools and integrated them and provided a little check mark if this particular area is highlighting a specific gene or a specific cell type. And we provided, and we called it a kidney genetic scorecard. And I hope you will use it. We had 32 different tools. That was up on the publication. Now we added 2 more, and hopefully after today's meeting, there are many additional tools to add. My vision for 20 years going forward would be that these tools essentially identify patients' risk or people's risk for kidney disease. We would present these sort of risk factors for people before kidney disease essentially develops, and then provide potential interventions that would be not based on traditional histology that is very, very hard to learn for patients and medical students, FSGS or collapsing, but based on the risk that they have for progression, or based on the risk of how we can correct that specific factor. If it's a gene, maybe we will correct it. If it's an environmental problem, we would avoid those specific issues. It could be metabolic or some sort of an environmental toxins. So I hope this is the way we would be able to go, but we have lots of challenges here.
So Julio, you tried to surface some of these challenges in the Basic Science Pre-Symposium on Artificial Intelligence. Do you want to update the listeners to what emerged from that and why you are so hopeful that more data might actually mean more knowledge?
Yes, and it is an important challenge, but one that we try to face with optimism. And I mentioned before these new AI tools that we can leverage, and this together with the expertise, but also the increasing data sets can bring us forward. And here, some of the challenge we discuss is on one hand that we need not only more data, but we need to improve the quality of the data. This has to be properly annotated. Also, we need to make sure the data is available and can be used by all, also following the principles of open science. And here, the KPMP initiative had really led the way with sharing the data with everyone. In the symposium, we went from the basic science all the way to the clinical context, and a key aspect is also, and I alluded this before, that we need to develop methods that are trustable. And for doing this, we need to properly benchmark them, to have the adequate ways to assess what we can rely them for and what we can really let patients or practitioners to work with.
Thank you. I hope you are as excited about where we are with Kidney Week 2025 and enjoy the next 2 days. There will be exciting insights. Some of the things which sounds like future is actually here. We will hear tomorrow about trials who are using these tools already today to see how one of our most life-saving drugs are working. So stay tuned on the next podcasts.